Hello, class. Welcome to Chapter 5, the Human Tissues. I'm Dr. Alendy, and let us get started. These are my resources and introduction. So, from Chapter 1, we learned what tissues are. A tissue is a group of similar cells and cell products working together to perform a specific role in an organ. We also learned what an organ is. An organ is a structure with discrete boundaries that is composed of two or more tissue types. Yeah, so this lab we're going to learn about our different tissues. We're also going to do a lot of histology. What is histology? Microscopic anatomy, which is the study of tissues and how they are arranged into organs. We're going to learn about our four different tissue types, and we're going to see there are different types and functions when it comes to their cells. They have a matrix and they have different amounts of space between their cells. Yes, you'll hear me say matrix and extracellular material. What is that? It is composed of fibrous proteins and a clear gel called the ground substance. What is that ground substance? It is also known as tissue fluid, extracellular fluid, interstitial fluid, or tissue gel. All right, so you'll hear me say these terms over and over again. Here we have our four primary tissue classes. We have epithelial tissue, connective tissue, nervous tissue, and muscular tissue. All right, what is epithelial tissue? Epithelial tissue, think of it as cells next to cells, next to cells, next to cells. It is tissue composed of closely adhering cells, and they cover your organ surfaces. They form glands. They serve for protection, secretion, and absorption. Where do we find this epithelial tissue? We find it in the epidermis, the lining of your digestive tract, in the liver, and other glands. Next, we have connective tissue. This is tissue with usually more matrix than cell volume. It is often specialized, so maybe you'll see me mention supportive connective tissue. There's a fluid connective tissue. There's a fibrous connective tissue. So you'll see all these different types of connective tissue. Next, we have nervous tissue. This is tissue containing excitable cells specialized for rapid transmission of coded information to other cells. So these are your nerve cells. So this is nervous tissue. So where do we find this? In your brain, your spinal cord, and of course your nerves. And then we have muscular tissue. This is tissue composed of elongated excitable muscle cells specialized for contraction. And we're going to learn about the three different types of muscle tissue: skeletal muscle, smooth muscle, and then cardiac muscle. All right, let's get started. So whenever you see an image going forward, it's either going to be one of these four tissue types. Before we do that, we're going to do a little bit of embryology. So at some point in time, we were made up of three primary germ layers. The three primary germ layers give rise to an entire organism. So those three primary germ layers are the ectoderm, the middle layer, which is the mesoderm, and then the endoderm. So in the top right image here, we see our three primary germ layers, the trilaminar germ disc. Yes, ectoderm, mesoderm, and then the endoderm. Yes. Let's see what comes from these three primary germ layers. First, we have our ectoderm. It gives rise to your epidermis and your nervous system. So your skin, your brain, your spinal cord, it comes from your ectoderm. Next, let's jump to our mesoderm, which is the middle layer. This mesoderm or middle layer becomes mesenchyme, and mesenchyme gives rise to cartilage, bone, and blood. Think of anything in the middle of your body, so deeper in your body, which is muscle. So which is middle in the body. You think of cartilage, bone, and blood. Mesoderm. Then we have endoderm, the inner layer. It gives rise to linings. So mucous membrane lining, digestive, respiratory tract, digestive glands, and many other things. All right, you need to know your 3 primary germ layers and what comes from these 3 layers. So whenever you were in lab, or whenever you will be in lab, you will see different slides. On those slides, you'll see CS or LS. What does that mean? Those are the different histological sections that were taken. So say you're taking a look at this blood vessel. If you cut it in the longitudinal section, it would look like this on the slide. That same vessel, if you cut it in the cross section, it would look like this on the slide. And if you were to cut it in the oblique section, it would look like this on the slide. Same vessel, it will look different on the slide. All right, so keep that in mind. When people in lab were taking their time to prepare your slides, they were using a fixative that prevents decay and different stains to help us to visualize different structures. All right, good. So we're starting off with our first class of tissue, epithelial tissue. These are closely adhering cells, 1 or more cells thick. So once you see cell, right next to cell, right next to cell, right next to cell, epithelial tissue. It covers body surfaces, it lines your body cavities, it makes up your glands, and it is avascular. What does avascular mean? A means without, vascular means without blood vessels, so it does not have any blood vessels. It lies on a layer of loose connective tissue. So in this image here, this is the epithelium. Here you see the connective tissue beneath. And your epithelium sits on a basement membrane. This is a layer between the epithelium and the connective tissue. So in this image here on the bottom, we see an epithelium sitting on a basement membrane. Epithelium sitting on a basement membrane. Epithelium sitting on a basement membrane. So how do we name our epithelium? Well, we first look at the number of layers. If there is 1 layer, we call that simple epithelium. If it is 2 or more layers, we call that stratified epithelium. And then we note the shape of those cells. And which cells do we name them by the shape of the apical layer of cells. So the layer of cells that is the free surface layer of cells. So this is the only layer. So this is the free surface layer. The shape is columnar. So this is simple columnar epithelium. Again, here in the bottom left, 1 layer, so it is simple, shape columnar, so it is simple columnar epithelium. In this one on the right, we have more than 1 layer, so it is stratified, and the shape of the apical layer of cells is squamous, so this is stratified squamous epithelium. Even though this layer that sits on the basement membrane is cuboidal, but the apical layer is squamous, so this is stratified squamous epithelium. All right, that is how we name our epithelial tissue. So let's go through the different types of simple epithelial tissue. So here we have 1 layer of flat squamous cells. So it is a simple squamous epithelium. Because it is 1 cell layer thick, it helps with rapid diffusion of transport of substances. It also secretes serous fluid. So where would you find this? Where do we need diffusion of substances across 1 cell layer thick? Think of your lungs. So in your alveoli, we find this. In the glomerulus of your kidneys, we find this. Your endothelium. What is your endothelium? That is the lining of your blood vessels. You can see why it also needs to be 1 layer thick. It allows oxygen and CO2 to diffuse across that endothelium, that simple squamous epithelium. And also your serosa. We're going to learn about serosa at the end. Your serosa is also made up of simple squamous epithelium. Next, we have simple cuboidal epithelium. How many layers? It is simple. So one, the shape you will see is cuboidal. And when we say cuboidal shape, we mean kind of a layer of square or round cells. Major function is absorption and secretion, mucus production and movement. And where do we find this? We find this in the liver, the thyroid, the mammary and salivary glands, bronchioles, and kidney tubules. In this image, this is a kidney tubule where we see one layer of cuboidal cells, so simple cuboidal cells. How do I know this is one layer? Again, we see a free surface, so this is the only layer. So this is that layer, and you see the harsh pinkish line beneath. That is your basement membrane. Again, here you see one layer of cells, the free surface here, and they are sitting on a basement membrane. All right. Also here, one layer of cuboidal cells, simple cuboidal epithelium. All right. Next, we have simple columnar epithelium. One layer of columnar shaped cells. The cells are closely adhered to each other. They often have an oval nuclei. The oval nuclei is often near the basal side of the cell. Simple columnar epithelium, it can have the brush border of microvilli. It can also have cilia, and they can often have also goblet cells. Here we see a goblet cell, goblet cell, goblet cells. Goblet cells secrete mucus. So those are mucus secreting cells. So here we have simple columnar epithelium on the left with goblet cells, and we also see microvilli. Here on the right, this is a unique type of epithelium. This is pseudo-stratified columnar epithelium. Why is it called pseudo-stratified? Pseudo means fake, stratified. So let's take a look at the drawing first. So here we see, let's focus on this strangely shaped cell. This strangely shaped cell, it has a somewhat columnar shape, but you can see it tapers out and it still touches the basement membrane. All of these cells, they still all touch the basement membrane. So it looks multi-layered, but all cells touch the basement membrane. This is pseudo or fake pseudo-stratified columnar epithelium. What else do we see? We see these cells here, and here those are goblet cells, and we see cilia. So this is ciliated pseudo-stratified columnar epithelium with goblet cells. So pseudo-stratified, it looks multi-layered, but all cells touch the basement membrane. There are nuclei at different levels. Yes, nuclei at different levels. It has cilia and goblet cells, and so the cilia help propel the mucus along. And where do we find this pseudo-stratified columnar epithelium? We find it in the respiratory tract. This one is taken from the trachea and portions of the male urethra. Yes? All right. Pseudo-stratified column. It's a special type of epithelium. Next, we're going to take a look at stratified epithelium. So, how many layers are there? More than one. Yes, two or more layers sitting on the basement membrane. Again, these are cells right next to each other. And we have three stratified epithelia named for the shape of their apical surface cells. We can have stratified squamous, stratified cuboidal, or stratified columnar. So here we have stratified squamous, stratified cuboidal, stratified columnar. There's also a fourth type called urothelium, also called transitional epithelium, which we're going to talk about. The deepest layers of stratified epithelium, it undergoes continuous mitosis. The daughter cells push towards the surface and become flatter as they migrate upwards. Eventually, they reach the surface, and then they flake off and become dust. And we can have two kinds of stratified squamous epithelium. keratinized and non-keratinized. Keratinized is found on skin surfaces, helps us resist abrasion. Non-keratinized, it lacks the surface layer of dead cells. So think of your skin. Your skin is made up of stratified squamous epithelium. We also learned that this epithelium is avascular, so it gets its blood vessels from the connective tissue it sits on. So as these cells migrate up and are pushed towards the surface, they get further away from the blood supply. They then start dying off and they become filled with keratin. So it is keratinized, killed cells. They do not have a nucleus. They can undergo, or they don't have a nucleus, so they are dead cells. Those are keratinized. Versus your esophagus also has stratified squamous epithelium, but so those cells are still alive. So you will still see nuclei in those top layers of cells. So let's take a look at it again. So we have on the left stratified squamous epithelium that is keratinized. And on the right, we have stratified squamous epithelium that is non-keratinized. So non-keratinized is non-keratinized here. These top layers of cells, when we make the slides, we often get these empty-looking cells, but we still see these nuclei. So these cells are alive. So they are non-keratinized, stratified squamous epithelium. Where do we find it? We find it in the tongue, the oral mucosa, the esophagus, and the vagina. Next, we have keratinized stratified squamous epithelium. Keratinized means dead-celled, killed cells. Multiple layers of cells become flat and skinny towards the surface. It is also there to resist abrasion, help prevent water loss, and therefore it helps to protect us. Where do we find it? The epidermis, and the palms and the soles are heavily keratinized. Next, we have stratified cuboidal epithelium. So here we have the epithelium here at the bottom left and then the top left. Stratified cuboidal epithelium, 2 or more cell layers. The surface cells are square or round. They secrete sweat, produce sperm, and produce ovarian hormones. So therefore, where do we find them? Sweat glands, ovarian follicles, and the seminiferous tubules of the testes. And then we have another special epithelium, which is urothelium or transitional epithelium. This is a multi-layered epithelium with surface cells that change from roundish, dome-shaped, and they can flatten out when stretched. Think of your bladder. When your bladder is empty, it's empty and it can therefore be filled with urine. As it fills with urine, this transitional epithelium can flatten out to allow for more filling of the bladder with urine. Yeah, so where do we find this? The ureter and the bladder. All right, and you will often see these dome-shaped cells. Yes, these dome-shaped cells. That is how you know you have transitional epithelium. This is an image I found from Draw It to Know It. Not sponsored, but I really like their summary slides, and I got this from their website. So a nice summary slide here. Next, we have connective tissue. Connective tissue, a diverse, abundant type of tissue in which cells occupy less space than matrix. Most cells are not in direct contact with each other. First, as epithelial tissue, we have cells next to cells, next to cells, next to cells. Here in connective tissue, most cells are not in direct contact with each other. They support, they connect, and they protect our organs. Some connective tissue has high vascularity, some have no blood vessels at all, so it depends. And here we have the functions of connective tissue. They connect our organs, so think of tendons and ligaments. They're there for support, so we have supportive connective tissue like bones and cartilage. They're there for protection, immune protection. Think of your blood. Your blood is a fluid connective tissue. For movement, storage, think of your adipocytes, where they store fats. Heat production, your muscles, your heat production, not your muscles. They're there for heat production, so adipocytes. brown fat for it, where we help with heat production, and again your blood or fluid connective tissue, allowing for transport of oxygen, CO2 in your white blood cells. So let's start off first with fibrous connective tissue. This connective tissue has cells and fibers. What cells will we find? You will find often you will find a lot of fibroblasts. These fibroblasts produce fibers and the ground substance of the matrix. You will also find our leukocytes or your white blood cells. You will find neutrophils, lymphocytes, macrophages. You'll also find plasma cells which synthesize antibodies. You'll find mast cells, and you'll find adipocytes that store triglycerides. So these are all the different types of cells you can find in connective tissue. You'll also find 3 different types of fibers. You will either find collagenous fibers. They give flexibility, they are stretch resistance. You'll find them in tendons, ligaments, deep layers of the skin. You'll find those collagenous fibers. You'll find reticular fibers, thin collagen fibers coated with glycoprotein. They form the framework, so please keep in mind, framework of the spleen and the lymph nodes and your tonsils, for example. And that framework is important, and they often stain very, very dark. And then we have elastic fiber. They are thinner than collagenous fibers, made of the protein elastin. It allows for stretch and recoil. And when it comes to that ground substance produced by the fibroblast, that ground substance is made up of glycosaminoglycans, proteoglycans, and adhesive glycoproteins. All right, we're going to start off looking at the fibrous connective tissue. When it comes to our fibrous connective tissue, we can have loose connective tissue, and we can have dense connective tissue. Loose connective tissue means there is a much gel-like ground substance between the cells. Dense, the fibers fill the spaces between the cells. Loose connective tissue can either be loose areolar or reticular. Dense, we can have dense regular and dense irregular connective tissue. Before we continue, we need to know what tendons and ligaments are in preparation for all the bones we will learn in chapters 7 and 8. So, a ligament connects bone to bone. A tendon connects muscle to bone. Yes, please know these two. All right, starting off with areolar tissue and reticular tissue. These two are examples of what? Loose connective tissue. All right. So, areolar tissue, we have loosely organized fibers, abundant blood vessels, many blood vessels. Where do we find it? Beneath our epithelium, in our serous membrane, underneath our serous membranes, between our muscles. So, areolar tissue is a type of loose connective tissue found underneath that basement membrane of your epithelium. So, the blood vessels that reside here, they have oxygen and blood, and oxygen is able to be fused up towards the epithelium. So here we have an example of areolar tissue. Doesn't it look airy? There's a lot of space between the cells and fiber. This is areolar tissue. These thicker pink fibers are collagen fibers. The thinner ones are elastic fibers, and these cells are all fibroblasts. Yes? Loose areolar tissue. Next we have reticular tissue. Remember I mentioned the term framework. They form a framework or a mesh of reticular fibers, and they stain very dark. They form the supportive stroma of your lymphatic organs, that framework. So you find them in the lymph nodes, the spleen, the thymus, and the bone marrow. Next we have dense connective tissue. Dense, we can have dense regular, a nice regular pattern here, versus dense irregular. There's no pattern at all. With dense regular connective tissue, we have densely packed parallel collagen fibers, and the fibroblast nuclei are compressed. Where do we find them? In your tendons and your ligaments. Then we have dense irregular connective tissue. densely packed, randomly arranged collagen fibers with very few visible cells. They're there to help withstand unpredictable stresses. Where do we find them? Deeper layers of the skin and the capsules around our organs. Amazing. Then next we have another type of connective tissue, your adipocytes. So your adipocytes are your fat cells. Yes, fat is the body's primary energy reservoir, and we have two types of fat cells of adipocytes: white fat and brown fat. White fat is the main type found in adults. You will see in the histology image it resembles chicken wire. It looks like empty cells. It provides thermal insulation. It cushions organs such as the eyeballs and the kidneys, and it contributes to the body contours of the body. When we get to the end of the semester and you'll dissect a cow's eye, you'll see that the optic nerve of the eye is surrounded by a lot of adipose tissue. It shows you how it's there to protect the optic nerve. All right? Brown fat found in fetuses, infants, and children. The color comes from the blood vessels and the mitochondrial enzymes, so it functions as a heat-generating tissue. So here we see that white adipose tissue. It looks like empty-looking cells. The nuclei are compressed against the periphery of the cell, but this looks like chicken wire. This is white adipose tissue. Next, we have cartilage. We saw cartilage. So cartilage is a stiff connective tissue with a flexible matrix. It gives shape to the ear, the tip of the nose, and the larynx. Cartilage is produced by cells called chondroblasts. Chondroblasts are not the smartest cells, all right? Your chondroblasts, they are cartilage cells that produce cartilage, the matrix. And when they produce this matrix, this ground substance that makes up the cartilage, they become trapped. And when they become trapped, now we call them chondrocytes. And they become trapped in a little cavity called lacunae. So chondrocytes are cartilage cells that are trapped in their lacunae, trapped in their cavities. Cartilage is covered by perichondrium. This is a sheet of dense, irregular connective tissue that surrounds elastic cartilage and most hyaline cartilage, but not articular cartilage. Cartilage is avascular, no blood vessels. Think of, for example, if someone gets a rhinoplasty and they have all this cartilage on all this cartilage of the nose, it takes a while to heal because it's avascular. So how does it get its nutrients and its oxygen? Through diffusion, which helps bring nutrients and removes waste, and it heals slowly. I like to also give the example of my dad. Whenever he shaves his head, he sometimes nicks his ear, and again it takes a while to heal because it is elastic cartilage, which is avascular. It heals slowly. The matrix of cartilage is rich in glycosaminoglycans, and it contains collagen fibers. And we have three types of cartilage: hyaline cartilage, fibrocartilage, and elastic cartilage. What is the difference between them? The types of cartilage vary with fiber composition. Let's have a look. We have hyaline cartilage, elastic cartilage, and fibrocartilage. By just looking at these three different types of cartilage, what can we see? We see a chondrocyte in its lacunae, but we see a little nest here where we have a bunch of chondrocytes in the lacunae together. Yes, we have a very lightly staining matrix. The background here, very lightly staining. This is hyaline cartilage. Hyaline cartilage is covered by perichondrium. So that layer there is the perichondrium. Next, we have elastic cartilage. We see a chondrocyte in lacunae, chondrocyte in lacunae, chondrocyte in lacunae. But what do we see in the matrix? We see elastic fiber. So this is elastic cartilage. Oh yes, this example of hyaline cartilage this one is taken from the trachea, from your primary bronchi here. elastic cartilage, this one is taken from the ear. And here we have fibrocartilage taken from your intervertebral discs. Fibrocartilage, we see chondrocytes and lacunae, chondrocytes and lacunae, chondrocytes and lacunae, but it's kind of a linear pattern to it, no? And we also see many collagen fibers in between the chondrocytes in the matrix. All right? So here we have all of that we just mentioned, but in words. Hyaline cartilage, clear, glassy appearance because of the fineness of the collagen fibers. It eases joint movement. It holds your airway open. It moves your vocal cords. It's your growth plate in your long bones. It's made up of hyaline cartilage, so it allows for growth of juvenile long bones. So where do we find it? Articular cartilage, costal cartilage in your trachea and your bronchi, the larynx, your voice box, and the fetal skeleton. What is articular cartilage? So do we just have, think of, for example, your femur sitting on top of your tibia and your fibula. Is it just bone on bone? No. At the end of your femur, you have articular cartilage. So also on top of your tibia and fibula, you also have articular cartilage. At the head of your humerus, you have articular cartilage. So you find this cartilage at the ends of your long bones, helping with ease of joint movement. Costal cartilage, where do we find that? On the ends of your, hopefully we're all yelling, ribs. Amazing. So yes, hyaline cartilage. So hyaline cartilage has perichondrium except for articular cartilage. It does not have perichondrium. Next we have elastic cartilage. Cartilage containing an abundance of elastic fibers, also covered by perichondrium. It provides flexible elastic support. Think of your tip of your nose, external of your ear, and your epiglottis. And then we have fibrocartilage. Cartilage containing large, coarse bundles of collagen fibers. Is there to resist compression and absorb shock. Where do we find this? The menisci of your knee, your pubic symphysis of your hip bones, and between the vertebrae of your spinal cord, your intervertebral discs. Next we have bone, also a type of connective tissue. Bone, specifically compact bone, is calcified bone arranged in layers. Those layers, we call them lamellae, and these lamellae are arranged around a central canal. So a central canal, which I'm encircling here, and its surrounding lamellae, we call those osteons. So bone is made up of osteons. So this highlighted portion here is an osteon, this is an osteon, that is an osteon, that is an osteon, a central canal, and its lamellae. And what do we see in between these lamellae? They look like little bugs. A zoomed in image here, those aren't bugs. No, those are osteocytes in their lacunae. Osteocyte in lacunae, osteocyte in lacunae, osteocyte in lacunae. These osteocytes were osteoblasts. Similar to the chondroblasts, they're not the smartest. Your osteoblasts, they secrete bone, the bone matrix around them. They become trapped, and then now they are called mature bone cells called osteocytes, which reside within their cavity called the lacunae. You see that these osteocytes have these thin projections called canaliculi. The canaliculi are delicate canals radiating from each lacunae to its neighbors, allowing osteocytes to contact each others. And just like how cartilage was surrounded by perichondrium, bone is surrounded by periosteum, a thick fibrous connective tissue covering the whole bone. And what connective tissue do you believe this would be? Hopefully we're all yelling dense, irregular connective tissue. Next we have blood, which is a fluid connective tissue. It transports cells and dissolved matter from place to place. So we have cells, and then we have the ground substance, which is the plasma. The cells in blood, those are our formed elements. So those are our erythrocytes or our red blood cells. our leukocytes or our white blood cells, and there are platelets, also called our thrombocytes. All right, so blood is a liquid connective tissue, fluid connective tissue. Next, we have nervous and muscle tissue. So nervous and muscular tissue, they are excitable tissues. What does that mean? Excitability. It is the ability to respond to stimuli by changing membrane potential. What is membrane potential? The electrical charge difference or voltage that occurs across the cell membrane. We learned in chapter 2 that we have, when we place salts in water, they become electrolytes, and electrolytes are able to voltage can be transmitted in those different solutions of electrolytes. That is what we have within our cells. Yes, we can have electrical charge difference across our cell membranes. In nerve cells, changes in voltage result in rapid transmission of signals to other cells. And in muscle cells, changes in voltage result in contraction and shortening of the cell. So these are our nervous and muscular tissue. Starting off with nervous tissue. These are specialized for communication by electrical and chemical signals. We'll learn about that when we get to the last 3 chapters of the semester. We have what cells do we find in nervous tissue? Our neurons, our nerve cells. They detect stimuli, respond quickly, and transmit coded information rapidly to other cells. And we also have other supportive cells called neuroglia or glial cells. They protect and assist your neurons. So which cells do we find in the nervous tissue? Neurons or nerve cells, as well as glial cells. When you see a neuron or a nerve cell, you will see a cell body, also called the neurosoma. It houses the nucleus and other organelles. Radiating from the soma, you have these dendrites, these short branch processes. They receive signals, and you'll find a large process called the axon. It sends outgoing signals to other cells. All right, this is nervous tissue. Next, we have muscular tissue. We have 3 types of muscular tissue: skeletal muscle, cardiac muscle, and smooth muscle. Skeletal muscle, what can we see here if we take a look at skeletal muscle? Skeletal muscle, we see a long cell, and we see many nuclei. Yes, so that is unique to skeletal muscle. Multinucleated long fibrous cells. We also see alternating dark light bands. Those are striations, and that is what we see in skeletal muscle. Next, we have cardiac muscle. What do we see? We see branched cells. We see one nucleus surrounded by lightly staining glycogen, and we see between each cardiac muscle cell, we see these intercalated discs, only found in cardiac muscle. And then we have smooth muscle. What do we see? Also just a central nucleus, and we have a fusiform or spindle shaped cell. All right, let's now take a look at words. So before we go through the different types of muscle again, I have a question. Do you have conscious control over your skeletal muscles? Can you tell yourself to pick up a pen? Yes. So skeletal muscle is voluntary. We have conscious control over skeletal muscles. How about cardiac muscle? Can I tell my heart, okay, now you need to beat. Okay, beat again. Oh, now you beat again. We cannot. We would forget. We do not have conscious control over skeletal muscle. Cardiac muscle is involuntary. How about smooth muscle? We find this in the walls of our hollow organs. For example, can I tell my stomach, okay, now you need to churn your food. Okay, churn your food again. Okay, mix the food. Okay, now let's open this sphincter so we can allow food out. Do we have control over that? No. So it's also involuntary. So skeletal muscle is voluntary, cardiac muscle is involuntary, smooth muscle is also involuntary. Which ones are striated? Skeletal muscle is striated, cardiac muscle is striated, smooth muscle, it is not striated. Skeletal muscle is made up of muscle fibers, long, thin cells. Most skeletal muscles attach to bone. It contains multiple nuclei and it has striations, which are alternating dark light bands, and it is voluntary. Cardiac muscle, limited to the heart wall. Your cardiac muscle cells, we call those cardiomyocytes. They are short and branched with only a centrally located nucleus surrounded by glycogen, and cardiomyocytes are joined to other cardiomyocytes by intercalated discs. And then we have smooth muscle made up of fusiform cells. So those are spindle-shaped cells that taper off at the ends. Yes, and that is, we find them in the viscera, we find them in the walls of our hollow organs. And here's another table going through the 3 different types of muscle tissue we have. All right, next we have so those were all of our 4 tissue types. Yes, epithelial tissue, connective tissue, nervous tissue, and muscular tissue. We need to know all of those tissue types, their subtypes, how to identify them on histology. We need to know all of that. All right, we got this. Next, we have the types of cellular junctions. What are cellular junctions? They are connections between 2 cells. Most cells are anchored to each other or to their matrix. Your cells communicate with each other, resist mechanical stress, and control what moves through the gaps between them. So let's take a look at 4 of our cell junctions here. Here at the top of the apical part of this cell, we see our tight junction. Here we see our desmosomes. At the bottom here we see our gap junctions, also called communicating junctions. And at the base of our cell, we have our hemidesmosome. By just looking at this image, what do you think the function is of our tight junction? Doesn't it seem like it is preventing anything from getting in between our cells? Okay. How about our desmosomes? What do we think this function is? A zoomed in view here, we see these linkages, these J-shaped hooks. It kind of looks like it's preventing us from being able to pull our cells apart. How about our gap junction or our communicating junctions? They are made out of connexons. One connexon here, and each of these proteins are called connexins. And don't they form a pore? So maybe it allows us to communicate with each other and allow us to share, maybe have movement of ions or something else. Yes, those doesn't need to look like that, communication. And then we have our hemidesmosome. Doesn't it look like half of a desmosome? And doesn't it look like it is anchoring this cell to your basement membrane? All right, good. So I took my time to make this table, so I would think it is important. And here we see our 4 cell junctions: tight junctions, desmosome, hemidesmosome, gap junction, and communicating junctions. Tight junctions, zipper-like interlocking linkage between 2 adjacent cells by transmembrane cell adhesion proteins. What do? They seal off intercellular space, making it difficult for substances to pass between the cells. All right, tight junctions. Next, we have desmosomes, a patch that holds cells together, kind of like a clothing snap. It keeps your cells from being pulled apart. It helps resist mechanical stress. We saw those hook-like J-shaped proteins arise from cytoskeleton again, helping keep our cells together. We have our hemidesmosomes, half of a desmosome, that anchors basal cells of an epithelium to an underlying basement membrane. It ensures that the epithelium cannot easily peel away from underlying tissue. And then we have our gap or communicating junction. These are formed by ring-like connexons. One connexon is made up of 6 connexins, and they form and they create a pore. And through this pore, ions, nutrients, and other small solutes can pass between the cells. It's located in cardiac, smooth muscle, embryonic tissue, the lens, and the cornea. Yes? All right, so know your cell junctions and their functions. Know where you find them. So if I give you a blank image, know which one is which. And next we have glands. Remember when we learned about epithelium, we saw that a lot of our epithelium makes up our glands. We can have exocrine glands and endocrine glands. Exocrine glands, they maintain their contact with the surface of the epithelium by way of a duct. Endocrine glands have no ducts. They secrete their hormones directly into the blood. Back to exocrine. By maintaining contact with the surface, we mean surfaces can be external, so think of your sweat glands opening up to your skin, tear glands opening up again to your skin, or internal, pancreatic ducts opening up to a pancreatic glands opening up into pancreatic ducts. Yes, for example, so we can have internal and external. Some organs have both endocrine and exocrine functions, for example, the liver, the gonads, and the pancreas. And your endocrine glands, they secrete hormones directly into the blood. What are those hormones? Chemical messengers that stimulate cells elsewhere in the body. So let's take a look at this example here. Here we have an endocrine gland. How do I know that? I have these cells secreting their substances directly into the blood. Versus here, this is exocrine. I have these cells secreting their substances into the center and then into a duct which is in contact with the surface. Yes? Endocrine, exocrine. No duct has ducts. All right. And what is a gland? I should have started with that. What is a gland? A cell or organ that secretes substances for use elsewhere in the body or releases them for elimination from the body. It's usually composed of epithelial tissue in a connective tissue framework and the capsule. So when it comes to secretion from glands, you will see the term. When it comes to the glands, you will see terms like secretion. What is secretion? When we release a substance or a product useful to the body. Versus excretion, we release a substance as a waste product to get it out of the body. And we can have many different glands with many different types of ducts. I won't test you on that, but there are many different types of exocrine glands and their ducts, and this is how you would classify them. We can also have 3 types of secretions from our glands. So we have serous glands. They produce a thin, watery secretion. Think of your sweat, milk, tears, and your digestive juices. Mucous glands, they produce a glycoprotein mucin, which absorbs water and forms mucus. And we already saw an example of a mucus gland, which is our goblet cell. This is an example of a unicellular mucus gland. And we can also have mixed glands, which contain both serous and mucus cell types. And yes, so that's what you need to know. We also have 3 modes of exocrine secretion. We have merocrine or ecrine, apocrine, and holocrine. Merocrine or ecrine secretion means the cells secretes via exocytosis. So exocytosis, we learned about that in chapters 1, 2, 3, or 4, exocytosis. So here we see this mammary gland secreting milk, sugar, and proteins by exocytosis. Apocrine secretion is where cells release membrane-bound vesicles. They bud from the cell surface with a thin coat of cytoplasm and plasma membrane. So here we see apocrine, we're releasing membrane-bound vesicles. And then we have holocrine secretion. The entire gland cell breaks down and becomes the secretion. So here we have these cells breaking down and the entire content reaches the surface, and that is holocrine secretion. Please know your 3 modes of exocrine secretion. Yes, merocrine or ecrine, apocrine, and holocrine. Next, we have mucous membranes and serous membranes. Do you remember what our serous membranes are made out of? Hopefully we're all yelling simple squamous epithelium. So let's start with serous, the simplest form here. Serous membrane, also called the serosa. This is an internal membrane. It is simple squamous epithelium resting on a layer of areolar tissue. It produces serous fluid that arises from the blood. It covers organs and lines walls of body cavities. An example of this is your endothelium. It lines your blood vessels in the heart. Another example is your mesothelium. It lines your body cavities. So your pericardium, peritoneum, and pleura are all examples of serous membranes. They're all examples of serosa. Next, we have mucous membrane or mucosa. It lines passages that are open to the external environment. So, for example, it lines your entire digestive tract. It has sublayers. So we have the epithelium, which sits on a basement membrane. We then have the lamina propria, which then sits on a muscularis mucosae, which is made up of smooth muscle. What is the major function? It has absorptive functions, secretory functions, and protective functions. And they often have mucus-producing goblet cells. All right, so here we see a mucosa or a mucous membrane with its epithelium sitting on a basement membrane. The lamina propria, which is, we see all of this loose areolar connective tissue, and we see the muscularis mucosa. Just a question for the class. What specific epithelium is this? We see nuclei at different levels. All cells are touching the basement membrane. We also see goblet cells and cilia. So this is pseudostratified columnar epithelium with cilia and goblet cells. Awesome. Next we have tissue growth. So tissue growth means increasing the number of cells or size of existing cells. We can have different types of tissue growth. We can have hyperplasia, which is growth through cell multiplication. So we have 4 cells becoming 6 cells. Yes, it's hyperplasia. We also have hypertrophy, enlargement of pre-existing cells. So we have 4 cells becoming even larger, same 4 cells. And then we have neoplasia, which is development of a tumor or neoplasm, where we have 4 cells changing in shell shape, cell structure. It can either be benign or malignant. That is neoplasia. You can also have atrophy, which is shrinkage of a tissue through loss in cell size or number. This occurs in normal aging or lack of use. So if you've ever had maybe you broke your ulna and your head was in a cast, your muscle cells would have undergone atrophy. And then we have metaplasia, changing from one type of mature tissue into another type. So for example, we have simple cuboidal tissue of the vagina before puberty undergoing normal metaplasia, and then it becomes stratified squamous epithelium after puberty. This is normal physiological metaplasia. First, we can also have pathological metaplasia, where we have in the lungs pseudostratified columnar epithelium of the bronchi becoming stratified squamous epithelium. All right, these are different examples of metaplasia, changing from one mature cell tissue into another. So tissues can change type within certain limits. That falls under differentiation, the development of more specialized form and function by unspecialized tissue. We saw this earlier where we saw embryonic mesenchyme becoming cartilage and bone. Alright, so let's continue with stem cells. So stem cells are undifferentiated cells that are not yet performing any specialized function. They have the potential to differentiate into one or more types of mature functional cells. That's because these stem cells have developmental plasticity, the ability of a stem cell to give rise to a diversity of mature cell types. For example, Before we develop into those three to those three primary germ layers, we were a morula. A morula is a stage of human development. It consists of 16 to 32 totipotent stem cells. What are totipotent stem cells? Those are embryonic stem cells that have the potential to develop into any type of fully differentiated human cells. And we get them from cells of very early embryo. We also have pluripotent embryonic stem cells. They can develop into any type of cells in the embryo, but not accessory organs of pregnancy. So they can give rise to your placenta or anything like that. Where do we get them from? From the cells of the inner cell mass of the embryo, so from the blastocyst. Next, we have adult stem cells. These are undifferentiated cells found in mature organs. We have what we call multipotent. They have the ability to develop into 2 or more cell lines. For example, bone marrow stem cells can give rise to white blood cells, red blood cells, many different types of white blood cells. Yes, multipotent stem cells. Versus unipotent, they produce only one cell type. For examples, stem cells giving rise to just sperm cells. They only give rise to sperm cells. For example, stem cells found in the stratum basale of your epidermis only give rise to skin cells to those same skin cells. All right. Tissue repair. Damaged tissue can be repaired in 2 ways. We can regenerate. We can replace the dead or damaged cells by the same type of cells as before. And when we have regeneration, we restore normal function. Versus fibrosis, we are replacing the damaged cells with scar tissue. Scar tissue means it's just there to hold the organs together. It does not restore its function. So know the difference between regeneration, tissue repair, and fibrosis. We also have necrosis, pathological tissue death due to trauma, toxins, or infection. And we have infarction, which is sudden death of tissue when blood supply is cut off. Alright, please know these 2 terms. Gangrene is tissue necrosis due to insufficient blood supply, which usually involves infections. We have, for example, dry gangrene, common complication of diabetes. Wet gangrene, which is liquefaction of internal organs with an infection. We also have gas gangrene, usually from an infection of a sole bacteria, and they release hydrogen bubbles, giving you gas gangrene. And it's also called decubitus ulcers. Decubitus ulcers are bed sores or pressure sores. They are a form of dry gangrene from continual pressure on the skin. And in this image, you can see that where these ulcers develop is where we have bone in very close contact with the skin. That's why if someone is bedbound, they need to be turned, I believe, every 2 hours. But they have to be turned every couple of hours, every certain amount. They need to be turned to prevent the development of these decubitus ulcers. All right. And then we have apoptosis, which is programmed cell death. So normal death of cells that have completed their function and best serve the body by dying and getting out of the way. This is normal programmed cell death. These cells undergo blebbing and then they are phagocytosed or eaten up by your macrophages and other cells. Billions of cells die by apoptosis, and every cell has its own built-in suicide program. So when a cell undergoes apoptosis, it's very controlled, there's no inflammation, but versus necrosis, we often have cell swelling and inflammation and many other reactions. This is a flow chart that you should also find on D2L where I made a flow chart to help you to go through the tissue histology. You have your 4 main tissue types: nervous, muscle, connective, epithelium, and then I have epithelium in the bottom here. It guides you through how to identify, how to start with naming, with nerve, same with muscle and connective tissue. So please review, use this flowchart while studying. These are the end of chapter questions. I try to highlight the more important ones you need to be familiar with, but all of them are important. But these are the end of chapter questions. Please, after studying, go through them, try to answer them yourselves, and then see if you got those questions correct. All right.